Hey guys, let's start off the today's class, and also we're going to review the questions from you over the last few days. I think we got some troops of questions in our Slack. Okay. Before we move into that, can you hear me? So that is my first question to you. I mean, if you can't hear me, or if you can hear me, okay, so yeah, someone confirmed that. And also you see the screens as well, right? So that's all good. Okay, so we have some sets of questions. So one from Ji Young, I can translate onto for the international students. So her question was, the height or the shapes we have. Okay, so the height or shapes, I mean, the physical shapes can be differed by sex in the same family because of the sexual hormones in our body, and that could be a factor for variation within a family. I mean, that was her question. I mean, is there any possibility for this? And also, is that related to the evolutionary process we've been forced during the evolutionary process? And or is there any gene contributing to height on the X or Y chromosome, right? So the answer is that definitely the sexual hormones is impacting on our body development, and that's for sure. And also that can impact on our developmental process postnatally, and also in utero situation as well. So when Galton investigate the parent-child pair height distribution, when we discussed in the previous session, he also factorized the biological sex into the calculation. So he definitely adjusts the height by female and male, and looking at the correlation between parent and child height. Okay, that was one thing. And then the factors impacting on height, I mean, the questions about the factors impacting on height, we have lots, as we see in the previous slide, because heights are interact with many additive alleles, as we learned in the polygenic model, and that's one thing. So given that situation, I mean, it is hard to say we can, I mean, there's only one single factors determining the sex differences on the sex chromosome, right? I mean, definitely sex chromosome is one of that, but that is not entire, the contributor to the variation across male and females, right? So that's one thing. And I'm not sure which part I'm introducing in that topic, but yes, but yeah. I mean the discussion about the additive alleles in heights, we're going to come up in later sections soon. Okay, so the next question is, she asked whether tongue rolling ability is dominant inheritance or not, right? And I mean, the reason why she asked that is that she saw some people can roll tongue after some rounds of practice. So I mean, that's why she thought that the tongue rolling probably the consequence of the effort people have during their life. So and then she judged that probably other than genetic factors, there must be environmental factors on tongue lolling, right? So that is really interesting question, and that question grabbed me over the few days, because I never thought about the tongue lolling is dominant or not. And then the first thing I did was I searched Twitter, and I saw some people debating about whatever tongue lolling is dominant or not, and they said I'm dominant person, or I'm recessive person, and I mean that's the typical Twitter. So but anyway, I mean my short question is that there's no study for identify whether tongue lolling is dominant or not. I mean there was a study, or there were some studies about that, but there was just small scale study, and I mean we couldn't really replicate that result, and I mean that means that we couldn't really trust that result. So I mean we could do the tongue rolling study with the large scale whatever genomic technology or whatever the national scale study, but the problem is that no one wants to study that trait, because that is not really top priority in our public health, right? So the modern science, one thing I should tell you is that it is been doing by the support from the government or a company, right? In the short words that without money we couldn't study that, right? So why government wants to study, invest the money to the study, is that is the top priority in public health. But like the traits like tongue lolling, no one really care whatever you roll the tongue or not. Who cares that, right? So people want to study who are sensitive to COVID-19, or who are sensitive to AIDS, or who are the valuable in developmental process or something. That's the thing we can get money from government, but tongue rolling, no one wants to study that. So that means that we don't know the answer. But if you want to get some answers from whatever Twitter or some stupid web page, then you can say that it's dominant, but it's not. That's one thing. And then related to this, I look at some questions about sort of the same question as tongue lolling, which is the earlobe attachment. That is interesting thing, because I found some papers, and okay, new share. This is the study from 2017, published in American Journal of Human Genetics, which is quite, great journal. And the study by the 23andMe, this is the one of the largest company, genetics company in the world. And they look at the people with the genotyping data, about 74,000 people. And they look at the self-questionnaire about whether you have, attached earlobe or not, right? And then they find some genes, and the genes or loci, they found 49 loci, which are strongly associated with the earlobe attachment, right? So according to Mendelian inheritance, we cannot answer that whether, earlobe attachment is dominant or recessive, right? There's no way we can do that, right? So there are many genes, and also these many genes involved in earlobe attachments according to Mendelian inheritance, right? Polygenic passion. Yeah, sorry, not Mendelian, right? So, this is one reason why we care about the polygenic inheritance, why we care about the additive factors, blah, blah, blah. Okay, back to the Slack. Okay, and another question from Minsa. So the first question was when she derived the formula in page 18, the Q2 equal PR isn't clear. The reason is that Pn plus 1 and Pn is the same. Under the assumption that these two are the same value, then they can say that across the generation, all of the three genotypes we could say it's an under the equilibrium, right? So I mean, it looks a bit of circulation issue, but it's actually the hardest assumption in the developing his formula. So under the assumption that the Pn+1 is equal to Pn, that case we could say that the other genotype frequency is derived from the equation 1 and 2. So that means that the all of the genotype has the same probability combined from the previous generation, and that is the maintain, and that is the constant across the generation, and the 3 genotypes. That's why it's under the equilibrium over the generation. So that was the answer for the first question. And the second question was the SND is the modern meaning of allele, right? And that was the main message in this class. So that means that on the chromosome, the units of gene or the units of nucleotides, right? And which one is referred to allele, right? That case, the nucleotide is an allele, right? So somehow when you study the Mendelian genetics in your high school books or general biology or anything translated in Korean language, the concept of gene and allele is quite similar because they translate in the same meaning. But the gene and the changes in nucleotide is totally different things, right? I mean, that could be related to each other because, the nucleotide change can happen within a gene, but there are totally different units. And that is a good question because when you think about the allele in the modern sense, this is not a gene. This is the change in the nucleotide. But one thing I cautious, I mean, in the answer was that it is not a single nucleotide change. It is a series of changes in a nucleotide because there are different ranges of the genetic variants in our genome. So please keep in mind that, and we're going to deal with this issue in the later section. Okay, and the third question is also quite interesting to me. It's it grabs me over the days as well. So Minza's question is the Yu's question was about the short finger. I mean, it's hard to pronounce. I just say the short finger, and that is by dominant. And her question was whether I'm not sure whether I translate correctly or not, but whether this short finger is explained by a single gene, or you explain this by single gene because he didn't know anything about polygenic at that time, right? That was the question. The short answer is that I don't think you know the polygenic model at the time. That's for sure. And also I'm not sure you knew the concept of gene at the time. They know the concept of the inheritance, but that's not related to the gene. So that's one thing. So how they describe the short finger phenotype in early days, that was, I found brilliant the description about that. So in 1908 in Scotland, the physician Harry Drinkwater, he saw some people with short finger, and he was so curious about that, and then he sent some description to the famous man in British. His name is William Bateson, and then they thought about whether this inheritance is Mendelian or not, right? So that was the sort of the starting point about the short finger story. But one thing you have to keep in mind is that the short finger, or whatever the difficult pronunciation, is not developed by a single gene. It's developed by many different genes, and the questions can be rising in your mind is that how different genes can consequence the same phenotype in the end. right? But that things can happen in many situations. And that is quite, I mean, you probably see lots of that examples in a modern medical genetics. And studying why is the one critical topic. So I mean, I couldn't summarize into a single sentence, but we will see more examples on that. And that is quite, good starting point why we care about this, many genes come up with the same consequence at the end. So for students with curiosity, you can go to this link. This is the online curation of the Mendelian genetics, but it actually, including all other diseases as well. But you can see the genetic, manuals of the disease from this site. And this is the search results for the short finger symptoms. And you will find that more than 100 genes related to this phenotype. Okay, that was one thing. And the last question from Gayong, and that was the she pointed out my incorrect explanation during the Morgan's experiment. That is good because I mean, when I explained that sentence during the class, and I thought that, I mean, I didn't calculate that the chi-square statistics, but I just say that, I mean, this is the didn't follow the non-hypothesis, and then I said that this is the anti-examples of the Mendelian inheritance linkage, but the Mendelian assortment. But then what I'm saying that I thought, I mean, I could be wrong, but our fearless students pointed out that issue, and did the calculation and was correct, right? So I mean, it didn't reject that the hypothesis, so we could say that experiments actually following the Mendelian inheritance, right? So I mean, there was an examples of how we calculate the chi-squares, not about the anti-examples. Okay, that's good. And if you have more questions, please leave on our Slack and also put it into your words, then we can discuss further. Okay, that is good. Back to our class today. Okay, so where are we now? We are on the chromosome and sex. That is the fourth topic of the session 2, right? So we are still in the how human inheritance and heredity idea been developed over the time, right? So, and we are middle of the 1910s and 20, and we are move on to chromosome and sex issue, because this is the critical the discussion link between the Mendelian genetics to the molecular biology and Thomas Morgan's finding. Okay, so we're going to have the 3 topics today. So first one is the chromosome and genetic units on the chromosomes, because this is sort of the prototype of an allele in a modern genetics. And then we're going to discuss about the meiosis, because meiosis describing the Mendelian patterns in our inheritance. And the third one is the chromosome theory, because this chromosome theory is the starting point supporting the Mendelian model. Okay, so you have some chapters for your understanding. Okay, so the first one, chromosome. Okay, so when, I mean, think about the Mendelian genetics. So when Mendel proposed the inheritance model, he used the terminology called hereditary factors, right? So hereditary factors in Mendelian rules were proposed. At the time, no one knew the biological basis of the factors at the time, right? So because the, I mean, Mendel or other people assumed that, there must be factors, or there must be particles working in these distant, phenotypic differences between individuals, and that is working in, whatever segregation way, assortment way, or dominant ways. But there's no way we can actually see the biological basis of the factors working mechanism, right? So in modern sense, we can say that the hereditary factors are genes and alleles on the chromosomes. And this meaning is been evolved over the time, right? So as I explained in Minsa's question in the Slack, we in most recent days, we are seeing alleles are hereditary factors, but before the genomics area, we thought that the genes are the major hereditary factors, mostly in the molecular biology time around the 1917 and 18s, right? But anyhow, the genes and alleles, they are all located on chromosomes, right? Okay, so in 19th century, one thing we can note that is the advances in light microscopy technology and using the dye staining. So we can actually, represent different compartments with the colors or fluorescence, and this is the dyeing staining technology. And we look at those, color patterns or whatever staining patterns using the lens scales, right? So at the time, lots of things is developed for the, the lens technology, and as I mentioned, the light microscopy technology is one of that. And this is probably the one you felt bored during the general biology, 101 introduction chapter, because this is all part of the how microscopy technology developed for the biology study, blah, blah, blah. But actually, it's a great advancement in biology and also genetics, and it actually brought so many discovery in our cell system, especially the behavior of chromosomes during cell division, right? And then it eventually proved the presence of hereditary factors in Mendelian rules, right? So that's the thing. Okay, so what is the chromosome? So when we're talking about the chromosomes, we could divide it into the two different types. So one is the nuclear chromosome, and the other one is the mitochondrial chromosomes, right? So nuclear chromosomes is in the nuclear, right? So nuclear is the compartment within a cell, and that is containing the nuclear chromosomes and containing the genetic materials that is derived from the father and mother, right? And that is the source of the Mendelian inheritance and also the central dogma patterns according to the molecular biology, and that is the major, the working factory for our phenotype, right? But we also have lots of mitochondria in our cells. So not just one, we have many in a cell, and also this many is the different, the arrangement and different numbers and different, the working purpose according to different cell types. So neuronal cells or whatever, the kidney cells or heart cells, these have different composition of, and also different, the compartments of the mitochondria genome, and also this is working for the, some energies and also other activities, right? So mitochondrial genomes are different from nuclear genome because the nuclear genome has the 23 chromosomes in 23 chromosome pairs in our genome, as we learned in general biology, like autosomal chromosomes or sex chromosomes, right? But mitochondrial chromosomes is only in mitochondria, and this is not pair. This is one set, so we called it haploid, right? And this is following the maternal transmission, right? So that's why this is the way we tracking, which ancestry we are from our maternal, pedigree, right? But there are some debates on there could be a paternal transmission of mitochondria, but this is still debate, so we couldn't, definitely say. But if you're interested in that, then you can read on that. Okay, so one thing I should tell you is back to the nuclear chromosomes. We have autosomal chromosomes and sex chromosome, and autosomal chromosomes from number 1 to 22. So we called it, chromosome 1 or whatever, chromosome 2, chromosome 3, blah, blah, blah. And the sex chromosome is X chromosome and Y chromosome. And this sex chromosome determine biological sex in human. So that is also a bit of, the topic we have to, define and articulate, but the sex and gender is different, right? So that's the first thing. So when you study biology or when you study genetics, especially, these days we are dealing with the two types of whatever the terminology called sex in previous generation, because in Korean words we only have one things in our society, because we never really thought about that issue over the generation. But in English language, it has the sex and gender, and that is the different, the concept we have to perceive in many different contexts. And gender is more the constructive form from the subjective and also the individualized perception. So and gender is the constructed by, of course, by biology, but also the social experience and also social interaction as well. So when you're talking about the sex and genders, it's quite different concept we have to describe in biological studies. So when you collect samples about your study for your study, then you can ask that, what is your sex, right? And she or he could answer that, I'm a female, I'm a male, right? But that answer could be gender, right? Because this is the self-identity, right? But if you do the genetic study, then you have to know about the sex chromosome for calculating many allele frequency or haploid, blah, blah, blah. Then in that case, you have to measure that the X and Y chromosome, and that situation you can identify whether this individual is male sex or female sex. That is different things from gender, right? We have sort of the same, sort of similar topics as gender and sex, which is the ancestry and ethnicity, right? So when you study some, whatever the genetic disorder in United States, then you can ask participant, what is your ethnicity, right? So some people ask that, some people answer that I'm a white, or I'm a black, or something, right? But when we look at the ancestry pattern from their DNA, then we can accurately, calculate that whether this person from European background or South American or something. That is quite different concept. So what I want to say is you have to pick up the right word to describe each experiment, and also bring that message to whatever you're writing, papers or research or communication. That is the critical things in this society, right? So in these days, right? Otherwise, we have to take so many different exceptions, and we have to describe so many different phenomena. So we always have to think about the right word and the direction to embrace all the people in our society. Some studies, especially in genetics or biology, is quite prone to the situation of the discrimination. So so that's why I'm long talking about the words about the sex in the sex chromosome. So keep in mind that also I always come up with that issue in our exam. So please review that. Even though I say this, but still 2 or 3 students failed to answer that question. So in that case, I have no way to save those people. But anyway, okay. So back to the next terminology, which is the homologous chromosome. So homologous means that the having a similar position, structure, value, or purpose, right? So if you look at the chromosomes, as I mentioned, that this is the pairs of chromosomes, right? So if you look at the chromosome 1, it has a pair like this, right? And these two are homologous to each other. So we call the homologous chromosome, right? So before the replication, it separates into the each part, right? And this each part, they are quite similar. They are quite akin to each other. And that's why we call it homologous chromosome, because they are not identical, right? So we call the homologous chromosome, and they have approximately the same length and centromere position. But they could have different compositions because of the genetic variants, right? So keep in mind that that's why I keep calling, it's homologous or similar. And then the homologous chromosome has the sister chromatid, which is within their pairs, right? So sister chromatid is the identical to each other, right? So because there is the complementary to each other. Okay. So homologous chromosomes are a key concept in meiosis because they are separate and they under the recombination and they shuffle the genetic material and put it into the gametes and transmit to the next generation. So think about the inheritance situation. We have to start from the homologous chromosomes. Okay, and also homologous chromosomes carry the same corresponding loci. They control the same inherited traits because they are complementary to each other as the sister's chromatid. Okay. Not complemented. Complemented means that it has identically, the matching towards A to T or G to C. So homologous chromosomes is not complementary. Just forget it. It's actually matching the loci on the chromosomes, but they could have different, compositions because of the genetic variants or because of the, the large copy number variation or something. So they could have different compositions, but they have the same locus or loci, which are controlling the same inherited traits. Okay, so sister's chromatid. So when we look at the homologous chromosomes, it contains the duplex of two sisters chromatids, which are joined at the centromere, like this, right? So when it's on the replication process, it's separated into the parts, but it actually joins during the normal stage on the cells, right? So sister's chromatid, as I mentioned, that this is the two identical copies of chromosomes from a single parent, right? And the pairing of the homologous chromosomes therefore produce a four-stranded structures, right? I mean, this is quite obvious because we have two sets of the two homologous chromosome in a pair, right? So there is one set, and each set of homologous chromosome has two chromatids. So in a pair of chromosome, we have four sisters chromatid, right? And then depends on how homologous chromosomes forming the sisters chromatid, we call it metacentric chromosome and acrocentric chromosome. So metacentric chromosome, as you can see here, this is quite, the same shape and identical, the length between this and middle of the centromere, right? And they are quite, the same pattern. But acrocentric is, we have the shorter things on the above the centromere, and below the centromere has the longer parts. So this is the two different shapes of that. Okay, then when we're talking about the same chromosome, so which is let's say this is the chromosome 1, and this is probably chromosome 20. And when you see these two, they are homologous to each other because they are from the same chromosome 1. But if you look at these two pair, then we say that these are the non-homologous chromosome. They are they are from different chromosomes, right? One from chromosome 1 and the other from chromosome 2. So that case we called it non-homologous chromosomes. Okay, that is good. And how we see this chromosome shape, with our eyes, and the way we can look at the chromosomes is the karyotyping. So karyotype is a cytological representation of chromosomes. So karyotype is a way to visualize chromosome shape, structure, and behavior of chromosome during cell division, right? So in karyotype, autosomes in metaphase spread are rearranged systematically in pairs from the longest to shortest, and people put numbers from 1 to 22. So this is the classic, karyotype of chromosomes, and we can say that this is a chromosome 1, 2, 3, blah, 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 22, and X and Y, right? So this is, we do the staining, the chromosome, and we look at their behavior over the cell division, and we can describe that, okay, this is the chromosome 1 and this is chromosome 2, and they look like, metacentric, they look like acrocentric, and they are autosomes and they are sex chromosomes, something, right? So this is the way how we see the chromosomes. And one of the, famous method is the Gemstone staining, and this is the way we do the bending style patterns in our chromosomes. And this is developed in, I think it's 1960s, 70s, and people doing the staining the chromosomes, and people start looking at the chromosome behavior and activities in our cells. And that is one of the, the classic way to do that. And this is just described in our textbook, as I mentioned in the previous slide. And it visualized the chromosomes with the short arm and also the long arm, right? So short arm, we call it P, and this is P means the petit, and the long arm, we called it Q, and Q doesn't mean anything, it's not P, right? So P and Q is the complement to each other, so we just say Q, right? So why P and Q matter? Because when we, describing something on our chromosome, we use this notation, right? So standard conventions or notation for a genomic position, we use P and Q, right? So let's say ABO blood type gene. So, whether you have A, whether you have B, or whether you have O or AB, then in that case, the gene determining this phenotype, this gene is located on 9q34, right? Then we can easily say that, okay, this is on the chromosome 9 and this is the section of 34, right? Okay, and this is probably on here, right? So as I put on this example, and as you can see here, this is the little cartoon to represent the chromosome position. We call the idiogram, right? Idiogram is the pictorial reference point that is useful for locating the position of individual genes on chromosomes. So this is the entire chromosome 9, and centromere is here, and this is the P arm, and this is the Q arm, right? So P arm, we have bit of sections like P24.1, 9P23, 9P21.3, blah, blah, blah. And Q has the 9Q12, 9Q13, or something, right? We use this notation for other, genetic syndromes as well. Pigment syndrome, which we call PMS, that's on the 20q13 deletion, right? So this is another convention way we describing the genetic disorder. So what that means, PMS, so Pigment syndrome, is caused by deletion, where chromosome 22 position on Q13, right? So this is the convention way we put the genomic loci on a disease classification. Okay, and also if you are interested in looking at the idiogram, Then I introduced the Ucsd genome browser. And this is the fun thing. This is like, you know, the Google map for human genetics. So, I mean, if you are bored enough during the weekend, you can play here, you know, over the weekend. I mean, it's free, right? So, I mean, you can spend like 48 hours nonstop. So if you go to chromosome X and P22.2, and this is on here, right? So this position has the gene called ACE2, right? Do you know what is the ACE2 gene doing in our genome? Do you know where it's working? I mean, if you know that, you can leave the questions or on the chat. I mean, that is my hope. At least one person knows what is the ACE2 is doing in our biology. Probably some people search the Google or some people search the Naver. Oh, okay. So modulating the blood pressure. Yeah, that is a good question. That is a good answer. Sorry. Yeah. And what is doing? ACE2. Okay. ACE2. ACE2 for COVID, right? So yeah, that's right. So SARS-CoV. So ACE2 is the receptor of COVID-19. The yeah. So that's why, you know, if you go to genome browser, you click that. I mean, genome browser knows that. I mean, which gene people wants to see in these days. That's the ACE2. So they are bring us to this position. Okay. So if you want to go to other genes, okay. So which genes you want to go? My favorite, the SN2A. Then now we are on the chromosome 2, right? So chromosome 2 position, and you can look at it in here. Then you can zoom out. Then you can see there whether these genes are conserved in rhesus, mouse, dog, blah, blah, blah. And also other, you know, other informations as well. So you can play here, whatever you want. Okay, so back to our slides because we have to finish our class. So, okay, so examples of idiogram. So this is the examples of the genomic loci of autism spectrum disorder, which are the topics of mine. And there are many deletions and duplication genetic variants working in autism. So if you want to represent that situation, then you can put the idiogram of each chromosome. So from chromosome 1 to 22, and chromosome X and Y, then you can put the numbers on here, the loci position. Then you can say that, you know, which study is finding that loci in autism patients, right? So idiogram is working in that purpose. Okay, so elements in chromosomes. So first thing we can describe about the chromosome is the ploidy. So ploidy is the number of homologous sets of chromosomes in a cell, right? So every species has unique chromosome numbers and ploidy. So let's say humans are diploid, and I don't know many in animals and plants, but at least the zebrafish has the tetraploid, which has the 4 sets of the chromosomes. But humans has the, you know, 2 sets of the chromosomes, so we call it diploid. D means the 2, right? And the rat has 2, mouse has 2, honeybee has haploid, only 1 set, because honeybees are all female. There's no male. Okay, so humans are diploid 46 chromosomes, including 23 from mother, 23 from father. It's very hard calculation, but I mean, in the end, we can say that 23 and 23. And a typical cells in living human being containing 23, but 46 pieces of DNA. Okay, so let's say the chromosome number 1 from mother and 1 from dad, right? So, and father, so, and chromosome 2 also 2, right? I don't know. So why I bit stop on this is this is the sentence from the human genetics book as well. I'm little bit, uncomfortable with this sentence because okay, so this is the I'm rewritten the sentence because it in original text it was a whatever the not living, it was the typical human something, because the author, the Harvard professor, he wants to describe that the typical humans has the 46 chromosomes, and some few people has the chromosome abnormality, like Turner syndrome or something. They have the extra chromosomes in their genome, but I mean, we couldn't say that is the non-typical situation, right? So I mean, it's again, it's bit the thing we should think about how we describe the correct word for the each situation. So, but anyway, okay. So that was one. So human adult cells containing 2 complete copies of the human genome. Okay, so that is the easy part. Okay, so hereditary units on chromosomes. So we have the factors are located on the chromosome, and that is working in the inheritance situation. And we could say the 3 things for this unit. So the first one is the locus, and the plural form is the loci, right? And allele and genotype. The locus is a fixed position or physical position on a chromosome, and that can containing the alleles or genotype, and that is ultimately controlling the phenotype. And alleles is the alternative form of the same gene occupying in a given locus on a chromosome. So this is the same, the concept of the Mendelian factors. And genotype is the composition of the alleles, right? So that is the major 3 units on a chromosomes. And chromosome organization. So chromosome not exist as a the discrete entity, or not exist in a sort of certain fixed position. It actually dynamically change their shape in a different situation, different cells, different developmental stage, right? So and of course the chromosome containing the DNA sequence, and this DNA sequence is compacted by the histones, and that is compacted again as to form the chromosome, as we see in this cartoon, right? So in other words, we can say that each chromosome in a nucleus has a long and linear double-stranded DNA molecule, right? And then when the gene transcription happens, that it unpacks and it gives you the double-stranded sequence, and where we can do the gene transcription to express some proteins. And nuclear genome consists of 6 billion nucleotides in 46 chromosomes. And also, hard calculation, 3 billion nucleotides in 23 chromosomes, right? In a haploid situation, right? Because I mean, we are thinking the chromosomes in a diploid situation, but when it forms gametes, then it separates into the haploid situation. Okay, so when you look at the chromosome organization, when the DNA is packed around something we call the chromatin, right? So chromatin is the DNA of all eukaryotic chromosomes, is associated with the number of protein molecules in a stable ordered aggregate, right? So what that means that the DNA sequence is long and linear format, so it has to be maintained in a stable situation. So the organisms, especially eukaryotic organisms, they evolve to have some stable format, which are the chromatins, right? And within this chromatin, it safely contains the DNA sequence, and they only unpack that issue during the gene description. Otherwise, it has the keeping their format. And in that complex, it has the histone proteins, and different composition of histone proteins is working in this organization and properly, right? So there was the organization part, and also histones has many different the protein format and also the composition. And histones have the 5 major types, which is the H1, H2A, H2B, H3, and H4. And each one has the different functions and different working mechanisms in different types of gene transcription and different cell types as well. So looking at those patterns and looking at those dynamics, we called it epigenetics, which are not properly dealt in the sections, but also the major topics in molecular biology and also the future clinical evaluation. Okay. So we learned about the chromosome organization. Okay, and the next topic is the cells and chromosomes. So we compare 2 different types of cells. So first one is the somatic cells, which are the major part of our constitution in our body. And the second one is the germ cells, which are in our reproductive organs, right? Okay. So somatic cells are the diploid, and it has the 2 sets of the chromosomes complement, so which are total 46 chromosomes. And germ cells, as I mentioned, that are the gametes, so have haploid and also N complement. So that means that in human context is 23 chromosomes, right? And germ cells, I mean, obviously, you know that the eggs and sperms, I mean, no more highlight on this. Okay, somatic cells. Okay, somatic cells contains both homologous chromosomes, and somatic cells is mostly working in, maintaining our bodies and, shaping our bodies and also growth and development. And obviously these are subject to mitosis, right? And mitosis is the cell division process, and this is the ordinary somatic cell division process, and which are modulating our body growth and differentiation and tissue regeneration as well. So what I meant ordinary is that if that is happening in an abnormal process, it goes to cancers, right? So mitosis is perceived as an sort of the ordinary process in our body activities, right? And this is the probably quite familiar issue from your general biology, and also germ cells as well. So germ cells contains only one of the homologous chromosomes, so and they are subject to meiosis, right? So this meiosis is a fundamental process for inheritance, and we are more focused on this process because these are related to Mendelian factors and how we related to the inheritance is working in our population, right? So germ cells undergoes meiosis, and this meiosis reducing the chromosome numbers by half, and it forms the gametes, which are sperms and egg cells. And during this meiosis, the recombination is happening, and this recombination process is the source of genetic variation in our organisms, right? And we're going to discuss this further in this slide. Okay, so meiosis has the 2 steps. So meiosis stage 1 and meiosis stage 2. And this entire meiosis process reduces the chromosome half and also generating the gametes with the variable genetic background. So meiosis 1 process the genetic recombination between the prophase 1 and metaphase 1, right? And this recombination also called meiotic crossover. We're going to learn in the next session. In the next slide. And the later stage, it reduced the chromosome numbers half at the anaphase of the meiosis 1. So from prophase, metaphase, crossover between these 2, then the separate these chromosome pairs into the half at the anaphase 1, and it puts it into the meiosis phase 2, right? So during this, the prophase 1 and metaphase 1, the crossover recombination process increasing the variation within the germ cells. So think about the the meiosis process is not working in a one cell. It's working in many different cells in our reproductive organs, and the numbers of a number of the gametes produced during this, the reproductive process, that it actually increased lots of different versions of the cells, right? Different versions of DNAs, right? That means that the different variation across the gametes, right? And only one thing is picked up and fertilized during the pregnancy. Okay, that was the how we increase the sort of the variation within a meiosis process. So what is the crossover? So when you have the germ cells in a meiosis situation, the homologous chromosomes is not separate by half. It's actually separate, but during this separation, they are shuffled and they are crossover to each other, and within the similar position, they cut in, they joined again, and then they form the new types of chromosomes and gametes, right? And this situation we call the crossover, or we can easily say that the exchange of the chromosome parts like this, like when we play the card games, well, I mean, two homologous chromosome pairs, they are shuffle their parts in a proximal position, and then at the end, the gametes has the different formats or different compartments of the not compartment, different sequences of the chromosomes in a gamete cells, right? And this phenomena was first described by Creighton and McClinton in 1931, and they are seeing this one in I think this one in corns. I think corns, yeah. And then they saw that these materials is definitely increasing the genetic variation because they are not identical to parents, right? And that's why this variation is introducing some variability in the next generation, right? That's why our offspring is not identical to the previous generation, right? Okay, so the exchange of genetic material between non-sisters chromosome, non-sisters chromatid, and the events which produce the genetic recombination, right? Okay, so in a sperm, the inheritance of the sequences on the typical chromosomes, which are one homologous contains sequence inherit from his father, and the other one is contains homologous sequence from his mother. So let's say this is your father, and one from grandfather, one from grandmom, right? And during the sperm meiosis in your father, these two chromosomes are crossover, recombination, and it forms two different types of the chromosomes in his gamete, which is in his sperm, right? So even though you get the one homologous chromosome from grandfather or grandmother, it actually has sort of the shuffled and mixed form transmit to the grandchild, right? So this is why we have different variation across every generation, right? Okay, so that is good. And then the meiosis too is similar to ordinary mitosis process, except the chromosome number is 23 instead of 46 because it's the forming gametes, right? Okay, and the part of the same thing. Okay, so implication of meiosis in Mendelian inheritance, we can think about two things. One is the segregation, and the other one is assortment, right? Segregation is obvious because the alleles are segregated during the meiosis one, and then it transmits to the next generation. So Mendel segregation is working, it's ruled because of the meiosis pattern, and assortment is also working because the each alleles on the locus is shuffled and is randomly recomposed during the crossover, and also it segregate to the next generation. But most of the situation we could say that assortment is working, but some of the part means that some of alleles are not shuffled and not randomly assortment. So that case, we say that this is, didn't follow that assortment, and we call the linkage, and we're going to learn in the next session. Okay, but anyway, back to the meiosis process. We can think about two ways of reproduction. So first one is accessory reproduction. So the accessory reproduction is you can reproduce without parents, right? So without both parents. So only one parent can reproduce by themselves, right? And most unicellular organisms reproduce in this way. So, okay. So the number of offspring from these accessory organisms, we can easily calculate that times by a certain coefficient, right? So if you have the numbers of the organisms in the first generation, then you can times the coefficient, then you can easily calculate how many offspring you will get in the next generation. That is quite easy calculation, right? But asexual reproduction has the pros and cons. So the pros is the good things is the asexual reproduction takes less time and in producing more offspring, because they don't have to do the mating. They don't have to find mates, right? So we can say that this process requires less energy, right? But the cons is that the parents and offspring has the same DNA, which means that they have the same genetic material and they have less variation in the population. That means that if the environmental or external force working on that population, and they're going to be entirely removed by that force, right? So we could say that it is fragile to environmental changes, right? But sexual reproduction is different because we need two parents, and we transmit our genetic material half to the next generation, and that version is slightly different because of the recombination and crossover. So that means that there is a great chance of variation across the generation, and this great variation maintaining the population over the many generations as well. So pros and cons about this sexual reproduction is the pros is to increase the variation in offspring, and organisms is more protective to many environmental forces because of this genetic variation. So if we lose some individuals in our population because of the fragile whatever fragile to certain external force, but other remaining individuals can continue on their the population growth in their area, right? So definitely having a great variation is much more protective to lots of different environmental forces. But the problem is that it requires some energies on finding mates. So that means the more cellular energies and cellular activities and additional, the efforts on that. And also, it takes lots of time to have offspring for this process as well. Okay, so the last topic we can discuss today is the rediscovery of Mendelian genetics from the study of chromosomes. So as I mentioned that the Mendelian genetics proposed in late 90s, but it's entirely ignored by the mainstream society. But after a few decades, the few people working in cells, the cytologists, they are see the Mendelian patterns in cells, and also they are most of them are actually located on the German, so they are the German people can read the German language. I mean, that's the written in Mendelian books, right? So they are easily accessed to Mendelian inheritance idea. And these cytologists, they are re-evaluate the meaning of the Mendelian genetics, and they embody that idea into the the meiosis process as well. And as we see in a meiosis process, the Mendelian factors is working as Mendel hypothesis in his model. Okay, so and one major figure we can think is the Walter Sutton. He is the first person clearly argued that the chromosome theory of heredity, right? So it's clearly the formulate how meiosis is working and what the chromosomes meaning and how this chromosomes is working in Mendelian inheritance. And what he proposed in his theory was this. So chromosomes has individuality. This is linked to the assortment, what Mendel proposed in his model, right? So because assortment has to be, the alleles has to be independently working on the trait inheritance, right? So he saw the chromosomes has the individuality. So within these cells, there are lots of chromosomes in the cell, but each cells, they are not dependent to each other, right? They are behaving differently in a cell. So Sutton assumed that the chromosome have individuality, right? And that was one thing. And chromosomes occur in pairs with members of each pair contributed by its parents, right? So during the reproductive process and fertilization, he saw that the pair of chromosome, one from father, one from mother, and that it merged into the next generation offspring, right? So that is perfectly describing the situation what Mendel proposed in his segregation rule, right? That was one thing. Okay, and the distribution of paternal and maternal chromosomes in each homologous pairs is independent of each other. So that is the sort of the combination of first and second description. Okay, so Walter Sutton proposed his idea, but in the same time, Theodore Boveri also noticed that issue as well, because that 1902, 1903 was that time. So they are come up with the same idea, and they proposed the chromosome theory of inheritance that is widely accepted in these days as well, right? Okay, so summary of the today's topic. So we discussed about the hereditary factors proposed in Mendel's model on chromosomes, right? In the perspective of genes and alleles, right? And we're going to more detailed about the genes in the next week section. And then we also discuss about both Mendel's law of segregation and independent assortment is described by the behavior of chromosome during meiosis. So we talked about the meiosis, right? And also this meiosis in sexual organisms, we are talking the increase of genetic variation due to the recombination crossover issue during meiosis 1. Okay, so hopefully you link those ideas into the why Darwin talking about the diversity trait values in a population, and also why Mendel talking about the segregation and assortment, and how people discuss that issue in early times, and especially before the molecular biology, and how these things is linked to the next topics. Okay, so that was everything we can have today. And for further reading, I don't know why I put these chapters. This is the duplicated one, as I mentioned on the first slide, the chapter 3 to 5, the Hartle book. You can read the chapters, or you can read this the Thompson books. Also, I put this centenary of genetics, but describing the advances in cytology and why why this is critical in genetics, and this is quite fun. So hopefully you enjoy these articles as well. And as I mentioned, if you have any questions, please leave yours on the Slack, and also shape your idea and tuning your thought, and hopefully you will prepare. The first exams are coming in 2 weeks. Okay, so that's good, and hopefully you have a wonderful week, and see you next time.